All right. So today we're kind of talking about a lot of really big ideas in vertebrate evolution. And that's kind of the challenge when you talk about fishes and fish biodiversity. And one of the reasons I try to spend time on fish biodiversity is because a lot of times these organisms kind of get talked about in the same week and a half in a class like this so that we can hurry up and get to mammals. And then in your minds, you guys have cartilage and jaws and fins all mixed up. And so I like to spend a little time in there and talk about the really incredible things we know about vertebrate evolution via these fish. So this is one of those fish I showed you a couple weeks ago. It's an elasmobranch. It's a kind of shark. This is called a saw shark. It's not the same thing as a sawfish. It's got a nice rostrum covered with like teeth that go sideways and these big electrosensory barbels for like sensing stuff in the ground. But I want you, when you see something like this, to like look, think about the vertebrate synapomorphes, the chordate synapomorphes, the gnathostome synapomorphes, the chondrichthyan synapomorphes. They're all there in that kind of an organism. And so when you're going around in the lab today, you're going to be looking at so many organisms. And I encourage you, even if I don't ask you explicitly, be like, all right, I'm looking at this little trout. What things can I see? What things do I know are there? But all right, I wanted to kind of talk to you really today about hard tissues, which is an awesome thing in vertebrate evolution, and fishes are our best way to understand it, because by the time we get to us tetrapods and us mammals, these tissues are very constrained. They only do like one thing. So it's very cool to see them as all these different structures. So just like we did skeletons last week, we're going to do all the different hard tissues that vertebrates make today. So the composition, you guys have notes on your lecture, on your lab notes. So you can take notes on that if you want to. But of course, you're welcome to use your notebook. Those are for you. So there's 4 parts of the composition of vertebrate hard tissues. When you think of bone, I think a lot of people think of bone, they think of something that is very rigid and crystalline. We kind of talked about this last week. You saw a brief diagram. But like bone is a living tissue. It grows, it can repair itself, it's alive. And so you don't need to like write down all of these little things, and you'll have these slides on Moodle. But the first part that I want you to always remember that's in vertebrate hard tissues, or sometimes isn't, and that's important, are cells. And there's tons of living cells inside of different tissues. So let's just say we're talking about bone. There's cells that, anatomists call osteoblasts. These are the cells that make bone. I think you're going to pick up on a pattern here really quickly. There's cells called chondroblasts. These are the cells that like lay down cartilage. Odontoblasts are cells that make this tissue called dentin we're going to talk about. It's in your teeth. Another kind of cell, a meloblast, makes enamel. And you can see these same prefixes, osteo, chondro, odonto, bone, cartilage, tooth. These are just the words scientists use to talk about the very different kind of cells. So in bone, you have osteoblasts that make bone as like an embryo is developing, as your bones are growing. But as bone grows, and this is a feature of bone we're going to talk about, some cells get trapped in the bone. They are surrounded in the crystalline structure. This right here is a picture of a piece of bone cut and then put on a microscopic slide so you can see the microstructure. These big holes here are where blood vessels pass through, so nutrients can move around. Sometimes there's a nerve that goes in there. All these little tiny black spots right there, those are all little spaces where one of these osteocytes lives and keeps the bone healthy, keeps the bone around. If you break your leg, these are the little bones that start like fixing it back up again, which is really cool. The other kind of cell that's in there, but not as common, is osteoclasts. Osteoclasts are kind of the cells, they break down your bone. As your bone grows and changes shape, when you had growing pains when you were a kid, there's like things that are happening, new bone being made, old bone being dissolved back. So there's all these living tissues, these living pieces inside a vertebrate hard tissue. There's a reason there's a bone cell for breaking down bone, living in bone, and making bone. In cartilage, we're going to talk about in a second, cells make cartilage, and there's cells that get trapped in cartilage, but cartilage doesn't get repaired. If you really tear cartilage, it doesn't like usually heal very well at all. Other things can step in, but cartilage doesn't heal like bone does. The second part of every vertebrate hard tissue is some kind of mineral component. So there is a crystalline structure to bone. It's usually this chemical here, hydroxyapatite, that's what's in bone. But there's all kinds of other hard tissue, are the crystalline compounds that make the crystals in hard tissues. So other things you guys might think about are things like keratin, like your fingernails are kind of hard. That's like a protein structure. It doesn't have a mineral component. But there's other kinds of animals like mollusks that make shells, or a really tough scorpion or something that has a chitinous exoskeleton. Some of these things, like the shells in a mollusk, have mineral components. but others don't. And so it's very interesting to look at like the deep history of some of these structures. There's always an organic or protein component. So out in the structure of the crystals, most of the bone is made up of that mineral, if we're talking about bone. Collagen is the protein most common in bone. And so if you guys look at this histological slide of a bone under a microscope, all this like streaking that looks kind of yellowy orange because of how they're lighting it, those are collagen fibers. So living, kind of stretchy things that can hold on to the different crystal pieces and keep the bone together. So you have some flexibility there, right? It's really awful. You might see like a sports injury where someone's leg like really bends, doesn't break. That's due to something like collagen that has strength, but it can take on some tension, whereas like the mineral structure bone can't take tension, it'll break. And the last thing is that you guys probably know that most of your body by volume is somehow aqueous. It has water in it. And that's definitely true for like hard tissues. Even a bone, even the enamel in your teeth has some amount of water in it. So I just want to like lay out amongst all the vertebrate hard tissues, these are the major structures: cells, minerals, so like a crystalline structure, proteins, and water. So just to put all that together to talk about one of these hard tissues, to talk about bone, this picture here, most everything you're seeing is the crystalline hydroxyapatite, the mineral. But there's a lot of protein, and then all those little black cells are living things. And all amongst the cells and all between the cells and throughout even the crystalline structure, there's H2O. It's sort of aqueous. So it's a very complicated living thing. You guys saw this last week, and all I did was have you look at it for a second. But this is what we're talking about here. So holes where blood vessels and nerves pass through. As the bone grew up, these little cells got trapped inside, and they keep the bone alive, they keep the bone healthy, they keep the bone nourished. Your bones do all kinds of things. You guys probably know there's marrow inside your bones, your red blood cells come from your bones. All kinds of things happen within like your femur. And you don't think about that. You think about it as like the thing that you stand on. And so just to give you guys a little bit of a back bracing for these different structures, for bone, it's mostly mineral. About 70% of that is. 20% of that is protein, 10% of that is water. These are like the estimates, right? Because the cells, which are the fourth thing, are by themselves very, 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 very minuscule. They're just a really important part because they're what keeps the organ alive. So that's bone. I always start with bone because you guys have bone. You think of bone. But you guys just learned in class that bone isn't a thing that all vertebrates have. Only osteichthyans really have bone. So the trout and the goldfish and the human are the only things that you just drew on your phylogeny that have this kind of a tissue, a bone tissue. That's pretty wild. So I want to talk about the other tissues that are more ancient. Oh, well, no, I guess I was just about to say this. So you guys and osteichthyans, trouts, goldfish, humans, you guys have this bone called endochondral bone. The bone deep in your skeleton is endochondral. That was the feature you had. And what endochondral actually means is it was formed in cartilage before it ossified, before it became the bone that you know. And so you saw that picture in lab or in lecture, and you're welcome to go back to that. This is like a very cool way for you guys, I hope, to start to appreciate how a bone forms. So in like an embryo, so this could be like a human femur, when the baby is developing, there's like this blob of cartilage that is formed first, and maybe this will be the femur. And in like long bones, so like your femur, your humerus, bones on in your limbs, there's ossification centers at the top and bottom. We call these like the ends, the articular surfaces. You guys might see like on a dog bone, right? That has like this piece, and then the main shaft of the bone. There's 3 different places where living tissue invades the cartilage and osteoblasts start to lay down the bone. So you get this growing here. This is what's happening actually right here in the middle of these bones when you guys were teenagers and you had like a growing pain. Maybe you did. I had growing pains. That's that pain is like this bone being like growing more and more. And so as the cartilage is replaced by bone, you get a bone. But the thing that I think people don't realize, and I want you guys to take away, is that the cartilage is what's there first. That's called endochondral bone. That is the snake morphine for all osteichthyans. And so you can see here that there's like new bone forming, and then the bone is growing. So there's cells that are eating away at the bone, cells that are adding new bone, reshaping it. It's a very dynamic thing. Bony skeletons are super adaptable because they can change. Anyway, there's something crazy just to show you like I'm not going to go into that too much. I wanted to put that on there just so you guys can appreciate, like, here's what's happening inside your femur and all the things that are being made inside your femur. Because it's an organ in some ways, even though obviously it's a structural thing. All right. So that was bone. Just some things I wanted to say to you about bone. Another of these vertebrate hard tissues, and this time I put hard in quotes because it's not hard at all, is cartilage. Cartilage is very ancient. Cartilage is way before, all the diversifications we're talking about here in vertebrata. Cartilage is inherited by all vertebrates from our common ancestor. Unlike bone, cartilage is mostly made out of the proteins, mostly made out of collagen. There's a lot of water in cartilage. There are no mineral structures in cartilage, usually. Very few cells. These are the cells that would get trapped in the cartilage and keep it alive. So cartilage is really easy to tear and injure. You guys know cartilage very well as humans in a lot of your facial structures. Mammals have a lot of cartilage, soft, hard parts on the joints of your limbs, maybe, on your face, in your nose, and your ears. But you guys know that cartilage goes back really far. Here is a skull, but it's smushy, of something like a shark with 3 capsules on it, because you guys know that inside that skull there's a tripartite brain with a nose, an eye, and an ear lobe on that brain. You guys are going to see some shark skulls today that have cartilage. Something I want to say about cartilage, because I think it's really important, is cartilage usually has no mineral component. But you guys learned that for chondrichthyans, the cartilaginous fishes, one of the synapomorphies, the characters of that group, is they have that prismatic calcified cartilage. So unlike lampreys, unlike your nose, sharks and rays and ratfishes do have some mineral component happening in their cartilage. It's their own thing that only they evolved. So whenever you heard when you were little that, like, oh, sharks have a cartilage skeleton, it's not this kind of cartilage that's all smushy. Sharks aren't smushy. They've done their own separate kind of mineralizing. I put this on here because you can't have endochondral bone. Osteichthyans can't evolve and have this hyper-evolvable skeleton without cartilage existing first. So cartilage is all over the place. Sharks and rays have their own special cartilage. You have cartilage, and a lot of your bone comes from cartilage first. These last two are all about some of the most interesting probably pieces of vertebrate evolution, but I'm gonna like really bite my tongue because all the stuff you can actually see and have evidence for is from fishes that are 400 million years old, and this is not that class, so we're not gonna go there. Dentin is another really, really, really important vertebrate hard tissue. You have dentin in your teeth. The white part of your teeth is dentin. The only dentin in your body as a mammal is in the white part of your teeth. Just like the bone, really, dentin is mostly mineral, about 20% protein and 10% water. It's super similar to bone, but one big difference is that there's no cells living inside of dentin. So if dentin gets cracked, if dentin gets eroded, dentin can't repair itself. There are no cells living inside there. When you guys, so here's the tooth, human tooth, and here's this crazy looking fish from 400 million years ago. Its whole body is covered in basically dentin plates. Talk about that in a minute. Here's you guys, this is how you know dentin. Dentin is here, the white part of the tooth. Enamel, we're going to talk about in a second, is the outside part of your tooth. Has anybody else besides me had a cavity? Yes, so what happens? How do you get a cavity? I've been blabbing for a minute, like a long minute. So why don't you guys talk to each other for a second? How do you get a cavity? What happens?
Sugar doesn't, but sugar uses bacteria. That's what happens. Usually with a cavity, because the enamel gets worn away.
All right, anybody feeling like they're going to be a hygienist or something and they want to tell us how you get a cavity? You don't have to want to be a hygienist. Come on, just at ISU, there are a lot of people who are. Any volunteers? Yes, yeah.
Their enamel wears down and then eventually some bacteria will start living. Too.
So you get your enamel, there's an enamel cap. You guys are mammals. You have crazy special teeth. You have no idea how special your teeth are. Don't worry, we're going to talk about it. But I'm only bringing this up right now because dentin is spread all throughout early vertebrate evolution. It's one of the most important hard tissues around. It's almost a, well, I won't even say that. It's really important. But the only place you have it is in your teeth. And so, like I said, there's no living cells inside dentin. So dentin can't repair itself. If some bacteria, which by the way, here's how you get a cavity. You eat gummy worms, you drink Coke or whatever. And all that means is bacteria that are living between your teeth have food to eat. They like to eat those sugars. That's all it is. There's a little coral reef ecosystem of bacteria in between your teeth. Kind of cool biologically, kind of a problem because they have waste products. Those waste products can like eat away your enamel, which is not, they're not trying to do anything. They're not trying to get in there. It just happens. But then once your enamel is punctured, your dentin starts to get worn away. That's where the nerves and blood vessels are, and your teeth cannot repair themselves. Once they grow, that's it. They're just done. They're little pieces of china. And if you break them, they're broken. And so that's why we care a lot about cavities, because we're monkeys that live for 90 years. And once we lose our teeth, we lose our teeth forever. So we really care about cavities. But I think you guys, I want you to take away, the reason cavities are a big deal is because once you get one, it has to get filled. Your body's not going to fix it. That's why. There's no cells inside the dentin. The thing that I want to say to you now, because we're going to come back to this in a second, is dentin in you, in most mammals, dentin is only up here in the teeth. But in early vertebrates, a lot of them have these crazy armor exoskeletons. Those exoskeletons are made like this is a piece of that armor right there, cut in cross section. That armor has a pulp cavity, dentin, and like enamel on the outside of it. So this fish's like tail fin thing, side fin thing, or side scale thing, you put it in cross section, has the same pulp cavity, dentin, enamel structure as the teeth in your mouth. That's weird. That's crazy. Dentin today is really only in mammal, for mammals, like I said, and for most of the vertebrates on land, it's only in our teeth. One thing that's really cool about dentin is that it in your teeth, dentin is usually capped by enamel, and then it has a set structure. Most of the crazy teeth you guys might know about, like maybe elephant tusks, are cool because they are teeth that have lost their enamel. So this elephant tusk is 100% dentin. It always is growing up in the elephant socket. And so when the elephant was a baby, that's where the tip of the tusk is. And then as an adult, here's the tusk ever growing. And so elephant ivory, things like this are 100% dentin. Big mammoth tusk, that's all 100% dentin. So it's really only in mammals up in the teeth. But earlier, it's everywhere. I want to say that dentin, we're going to talk about the skeleton here in a minute, is derived from ectoderm. So you guys might remember that a bilaterian animal embryo has 3 tissue layers: an ectoderm on the outside, a mesoderm in the middle, and an endoderm on the inside that lines the gut. Those are one of the features that unite. I hope we're starting to be able to see these like crazy deep connections. Ectoderm is the outer layer of a bilaterian embryo. Oh, the dentin in your teeth comes from the outer layer, the skin outer layer from your embryological state. Get there in a second. Here's enamel. It's the same picture as you just saw for dentin. I did make the word hard bold, just so you know, maybe we'll notice that. So enamel is the 4th of the vertebrate hard tissues I want to talk to you guys about. Enamel is almost completely a crystal. 96% of the structure of enamel is the minerals like hydroxyapatite. It's like glass. It's really, really, really rigid. It can crack. There are no cells living in it. So enamel also cannot really repair itself. It can be laid down by cells. So there are amelioblasts, cells that lay down enamel, but there are no ameliocytes. There's no cells that live inside enamel. So again, only in you, it's restricted to teeth. It's super, super, super resistant in wear. But as you guys might expect, in a lot of early fish groups, and we're going to talk about it here like in sharks, it's all over their bodies in their scales. So something that we'll talk about later in class, there's a picture with enamel circled on the little early fish scale and then in your tooth. We'll talk about this later in class, but mammals have turned to the fact that their teeth are made out of both dentin, which is a little bit soft, and enamel, which is super not soft, into all kinds of adaptations. this is a beaver's molars. And all those different colors, the orange is dentin and the white is enamel. There's ridges of enamel. And then when the beaver's chewing grass and wood and stuff like that, its teeth wear down, but the dentin wears down faster and the enamel stays like ridges. So you get like cool tooth stuff happening because mammals especially start to play with enamel and dentin as these two different kind of structure things. Also derived from ectoderm, so only on the outside of the body. Okay, now I want to actually zoom in on the thing I was hinting at this whole time. So you guys learned that Gnathostomata, the jawed vertebrates, yes, they have jaws, they have pectoral fins, they have pelvic fins, but two of the other features I gave you were that they have a dermal like skeleton, a skin skeleton, aka like heavy duty scales on the outside of their bodies. And the other thing I gave you was that they have teeth. The teeth in the mouth of most of these animals and the scales on some of them, especially things like sharks, show us that these are all the same kind of structure. So this is a placoid scale. This is the scale that you find sticking out of a shark. Does anybody know any facts or pieces of information at all about like shark skin? Yes.
It's smooth in the anterior to posterior direction and then rough in the others.
So if you pet a shark, this is nice. This is not nice. Yeah, shark skin's really rough. Sharks have almost no parasites like attached onto them, like little ones, because they have this really intense skin. A lot of swimmers swim with like fake like swimsuits that emulate shark skin because the scales of a shark are so tiny and they move through the water so efficiently. What you might have heard, and raise your hand maybe if you have, is that sharks are covered with millions of little teeth. Anybody ever heard that? No? Well, they are. That's what's so crazy. If you cut into a shark skin, and again, do like a histological slide of a shark scale, there's a pulp cavity. It's mostly made of dentin, and then it's topped with enamel. That's weird. So if you like take a piece of skin from a shark's tail, all the little scales look like they're teeny little tiny teeth. And so what's really cool is we know in shark jaws that that structure of pulp cavity, enamel, dentin, that's what a tooth is. And so one of these really big questions in vertebrate evolution was always, how did we go from all these early fishes that have like dentin armor, maybe with some enamel on it, their skin is what came first, is covered with these toothy-like structures, to the gnathostomes that have their teeth, and fused to their jaws, there are these big sharp things that are derived from skin. It's really, really crazy. So I just want to make the point here again that if we take a shark's fin or shark scale from its body, there's a pulp cavity, dentin, and enamel, exactly chemically like yours. In your mouth, a pulp cavity, dentin, and enamel. Very, very, very weird. Very, very cool. And of course, there's a great story behind it that we won't talk about too much because it's paleontological. There's all those same structures across shark's teeth, human teeth, and shark skin. I think, I'm looking at the time and realizing we are going a little more than I wanted to. I'm kind of blabbing at you guys because I thought I'd save lecture time. Let's do one more thing here, and then I'm going to stop and let you guys do the lab. Yeah, we'll just do this part. So I just think this is really important and really interesting. So the outer, I want you guys to know where different parts of your skeleton come from embryologically. This is something that you're going to hear next week in lab. Dr. Ray, developmental biologist in the department, she's going to be giving a presentation about Evo-Devo and what we learn from like embryos about comparative anatomy. So this here, we're kind of jumping laterally here a little bit. This here is one of our deuterostome embryos. So the cells become a ball, and as that ball splits and gets a hole, this hole is becomes the anus, and the mouth comes second. So deuterostome is second mouth. So you and a starfish have a deuterostome embryo. That embryo has an ectoderm, an endoderm. So the endoderm is the gut lining, the ectoderm is the outer tissue layers, and then the mesoderm, which is where most of your organs come in. Those are the early tissue layers, the triploblast tissues of all bilaterians, and deuterostomes do it anus to mouth first, second. So in your skeleton, different parts of your skeleton comes from different places. We're going to not worry about these head parts right now, but I want you guys to know that things like armor, osteoderms. The dramatic cranium is most of the bones of your skull and your teeth. You'll see that on the slide from when I gave you osteichthyes. Comes from the ectoderm, the outer layer. The mesoderm is the middle layer, and all that comes from that I said where most of your skeleton comes from. Your appendicular skeleton, so your pelvic girdle, your pectoral girdle, your vertebrae, your ribs, and also your brain case. The ancestral vertebrate skull is a neurocranium. So the synapomorphy for vertebrates of skull comes from the mesoderm. And then I'm going to actually do this in lecture. The endoderm is part of this, from the endoderm is this part of your skull called the splanchnocrinium. That's the gill structure. I know that this is too much right now. So I'm going to pull up on this and get you guys into the lab. So you guys did the phylogeny. And what I'd like to have you do now is actually have some time with specimens. Maybe I'll come back to what I wanted to say after you guys get going. So your lab today is just trying to get you to look at all these different early vertebrates in terms of splitting from one another. There's osteichthyans, there's chondrichthyans, there's all kinds of stuff in here. They're spread out all around the classroom. I will get gloves out. The point of today's lab is to give you guys a chance to like touch and look at a bunch of these organisms, because I just give you pictures all the time. Do more drawing, do more interpreting. I'm going to put gloves around so you can look, but most of the lab is just focused on giving you the chance to like take characters, take observations, draw them, make them. I don't usually like to talk for 40 minutes. That was way too much. So I'm gonna let you guys get after it. I'll point what places are which once you guys start moving around. I'll put gloves out. This right here. All right, so just to get you guys a little bit more easier in the lab today, some of our early chordates, we've got tunicates and cephalochordates back here at this microscope. Here back in the middle, we've got wet specimens and dried specimens of hagfishes and lampreys through your cyclostomes. Over here, we're getting into the jawed vertebrates, and we have the chondrichthyans first. So there's a bunch of shark and stingray stuff here, and a bunch of shark and stingray and ratfish stuff up there. I brought you guys a megalodon too from the museum. I don't know if you have a cool shark. And then we've got our bony vertebrates here and there, our osteichthyans, our axonopterygians, all of which is part of the lab. So I will have gloves on. I will get specimens out of jars. You guys are welcome to put gloves on to like pick stuff up and look at it. Please be very careful with wet specimens. I borrowed them from the ichthyology people. But I really like the idea that you guys are going to get the time to like look at 4, 5, 6, 7 fish today and really just like peek. Look at your lecture notes. Let's find cool stuff to talk about. I hope this is like a fun. I'm gonna stop. I'm gonna shut up.
No.
You stole it. What did I go over? It's wait. We have one that's like way bigger.
Yeah, no, it's helium. Focus on. No,
There's one of the first. And I brought it. There's like a lot of.
maybe.
I don't know.
They're not.
That's
Or to the other.
You can see Radic, yeah.
They have.
Most important. That was.
I mean, just look at the roof of this building. They're divided.
These are the
in
things
parts.
that.
These are like the gill.
Yeah, that's like you can see the skull, I'll come around.
What is this?
This is the ratfish and the stingray. And actually a shark, too. Got a shark. I found this little one shark and I was like, why wouldn't I bring out this little
Not very things are lateral. And one of the curiosities to land on.
Well, yeah,
They keep going, so on a triple table.
But there's valid. Yeah, oh, good. I wasn't sure if it had everything I was in, but it does. Alright, let me get some stuff. Yeah, right.
All right, so there's definitely like stuff is spread out a little bit, so please do ask me questions about what you're looking at if you don't know, or talk to your neighbors. So like, there's actinopterygians here and actinopterygians here, you've got to look at both stations, even though they're completely different kinds of fishes. This is where you'll see the shark jaws with the gills articulated in these jars here. That's the only thing I'm asking you to draw, and there's a bunch of just like cool shark stuff. And then up there's a bunch of elasmobranchs. It only says ratfish and like a skate and a ray, but I also brought a little baby shark. It's not a baby actually, it's just a little shark, but that's cool. So you can go over and draw the shark if you want, then Lamprey hagfish at the back. Osteopean skulls are up by where I've seen.
skating and roll.